That was just like a really.
We were waiting. Okay, now, let's go through the story. All right. So this is how I suggest you do it when you are doing your feedback paper in a few moments. All right. So, and as I had said earlier, I'm hoping you're bored with this. All right. So what we've got here is we've got a piece of DNA, and it has inside it, incorporated into it, viral DNA. So let's just do the viral DNA in red. And Lana, you had asked that question earlier, and then you came to that conclusion. Yeah, it can do this because it is made out of nucleotides. And I had said, I think it was yesterday, that there's no chemical difference between viral DNA and your DNA. It's all the same chemical, it's just the order of the chemicals that makes the difference. But here, we have some viral DNA incorporated into this, and I had said that, so the reproductive strategy of such a virus as this, a lysogenic virus, is to let the cell copy it. And as the cell containing this DNA copies it, and you end up going from one cell to two through mitosis, and then down the line, each of those cells will replicate. What you would have is an exponentially growing viral infection. Devious. The virus does no work here. It just lets the cell do what cells do, and you end up with millions of, potentially millions of viruses in millions of cells, and then later on down the line, those viruses just decide to go lytic. Well, you could become very sick very fast if you just lost millions of cells all at once. But that is the style of this virus. All right, so how does it get copied? Well, first, what does it mean to copy? It means that at the end of the process, we end up with two pieces of DNA. We started with one piece, now we got two, and they're copies of each other. All right, and then each of these pieces of DNA would be packaged in a cell at the end of mitosis. So there's this thing at the end of mitosis. I always remember this, it was just something stuck in my mind. At the end of mitosis, you have this thing called cytokinesis, where the cell just kind of pinches in off of itself and divides into two. Well, the goal would be, this would go in one cell, this would go in another, and now you have two cells, and those two cells each have a virus in them. Whereas what we had to begin with was just one cell with a virus in it. So, okay, so how do we go from here to here? Well, the way you show that is you show this intermediate stage called a replication fork. All right, and so what the replication fork is it represents the fact that the DNA to be copied is opened up, unzipped. So we're going to run a zipper right down through here. I'll pull these two pieces apart, and then we're going to use each piece as a template to copy the DNA. So we'll pull this apart. So here the pieces are pulled apart. And then we're going to follow Chargaff's rules. A goes with T and G goes with C. And we are going to build there's a nucleotide, there's a nucleotide, there's a nucleotide, there's a nucleotide. We are going to use the top to rebuild the bottom piece, and on this DNA, we're going to use the bottom to rebuild what would be the top piece of DNA. So, anybody remember the term that's applied to that kind of copying? It also would be describe my political views. They're semi-conservative, where each new cell ends up with one piece of old DNA and then one piece of new DNA. All right, so now, once the DNA begins to be unzipped, we're going to start copying this DNA. We're going to start building the missing strands. However, we got to build them according to the 5', 3' anti-parallel nature of DNA. And I had said that I'll always make this corner up here 5'. And if that's 5' and DNA is anti-parallel, that means this is 3'. And then it means down here at the end of this top strand is the 3' end and 5' end. And so what I'm asking you to do here is reflect back onto the model you built last week. This is just laying on its side. So, okay, there's a rule here. The rule says that DNA can only be built from the 5' end to the 3' end. You can only build DNA by adding to the 3' end. All right, so if we look at the bottom, this will be the 5' end right here of the new top strand. Here, let's make the new piece blue. So that's a new nucleotide. And we put it there because we read this nucleotide and follow Chargaff's rules. So put that nucleotide in place. This would be the 3' end of that nucleotide. All right, now we're going to add the next nucleotide because we want to build a chain. Well, we would add it here. You always add to the 3' end, which means the next nucleotide would go here. So the cell's machinery is going to read that, whatever that is, whatever nitrogenous base is there, it's going to follow Chargaff's rules, get the correct nucleotide, and put it right there. And then it's going to go and it's going to read this one, and it's going to put it right there. It's going to read that one, and it's going to put it right here. And so what we're doing is we're using the old strand as a template to build the new strand, and we're building it in this direction, from the 5' to the 3' direction. All right, now, the mechanism that is unzipping the DNA is going this direction. And at least for the bottom strand, it is being built in the same direction the zipper is going. So what will happen is the cell will let that zipper get out there a little bit, then it's going to grab on here, and it's going to begin copying the DNA, and it's just going to go right behind the zipper. So as this is moving this direction, this becomes unzipped, and that's unzipped, and that's unzipped, and that, and eventually what we have is the entire piece is unzipped, and at least on the bottom strand, it just gets copied all the way through. So what you end up with is this down here. You end up with one long piece of DNA that's fully copied, and it is made of one piece. It's not busted into fragments. This is called the continuous piece. It is built continuously. And the reason it's built continuously is because the mechanism that's building this new piece of DNA here is following the zipper. They're going in the same direction. All right. But the story for the top strand is different. Here's the story for the top strand up here. You see, if this is the 5' end of the top piece, the top template, then any new DNA we build here, given that it's anti-parallel, this is the 3' end of the new piece of DNA that needs to be built. And you can't grab a nucleotide and put it here, and then grab a nucleotide and put it here. That's what we did below. That's because we would be building it by building it in the 5' direction. If this is the 3' end, then this end down here would be the 5' end. And DNA cannot be built from 3' to 5'. So this can't happen. Somebody asked in an earlier class, well, why does DNA have to be built by adding to the 3' end only? And it just is. That's a biological fact. I don't know why it is that way. So what happens is, to build this top piece here, the machinery of the cell has to start here, close to the zipper, and add the nucleotide. So it adds a nucleotide here. This would be the 5' end. This would be the nucleotide's 3' end. Then it reads this, and it grabs another nucleotide, and it reads this, and grabs another nucleotide. And so what it does is it builds that top piece this direction. And at the end, if that was just the whole story, at the end, what you would have is just this. You would just have this tiny little piece of DNA built. Because this couldn't be built because it hadn't been unzipped yet. Now, so what we get is just a tiny fragment on top, and as opposed to the entire piece on the bottom. And that fragment, what's the fragment called? It's an Okazaki fragment named after the lady who discovered it. And she also discovered how the cell solves this, because this cannot stay this way. You can't have whatever cell's going to get this DNA would have a busted up piece of DNA, and that doesn't work. So the cell can't leave it that way. Right? So what actually happens is as this piece is being built this way, the zipper keeps going. And eventually, this stuff will become unzipped as well. So like kind of my hands here, the zipper's going this way, but this is being built this way. Eventually, let's say the zipper gets down to here. What this will do is it pops off, comes over here, grabs on, and it builds a fragment until it bumps into the previous fragment. And then as it's building this way, the zipper keeps going, and eventually it will pop off and go over and build another fragment. So the top strand is constantly popping off and re-grabbing on, popping off, building a fragment, re-grabbing on. It's constantly doing this. And so what you end up building on the top is a series of tiny fragments. And those fragments will have to be welded together. So in the end, you would have two complete pieces of DNA. It's just this top strand it takes way more effort and more time for it to happen. And so what we say is the top strand is lagging because it takes extra time and more effort for this whole popping off, reattaching, popping off, reattaching, popping off, reattaching. All right, so third time. Boring? You got it? Okay, now what I will add to this is this entire process would be unnecessary if the DNA could get completely unzipped first. So if the DNA got completely unzipped first, what you'd end up with is like zzz, it's getting unzipped. Zzz, there it is, it got unzipped. Here's the virus in there, it got unzipped as well. And if it could get unzipped completely first, then this could build this direction continuously, and then you could grab on here and build this direction continuously, and both would get copied. But that's not what happens. And I think the reason it doesn't happen that way is because on a molecular level, DNA is enormously long. And if you had to wait and for the whole thing to get unzipped, then the cell would be sitting there waiting for I don't know, an hour or so while all the DNA got unzipped. And by doing this, by making these fragments, it actually is faster to do it that way because it doesn't have kill time waiting for the whole thing to unzip. Somebody in here, I think it was this class, was asking me about this subject. Was it? Do you guys remember asking me about this? About like this very thing. Was why doesn't it just unzip the whole thing? I think that was you. Was that you, Elena? Okay, so that was. And then I said this gets even more complicated for bacteria because bacteria DNA is in a circle. They don't have chromosomes. They have a big loop of DNA, and there is no end on a loop. So you can't start and then wait for it to get unzipped because it's in a loop. There's no end to start on. So you're always, well, is that true? I guess you could unzip in a circle and have two circles pop off. I guess that's true. I guess it may be the same problem. It just doesn't do it. It takes too much time to wait for the whole thing to get unzipped. Okay, so now it's time for feedback.